Right, good morning, everyone. Let's get started. Let's finish cycle 3 with inheritance of sameness. The focus will be on the S phase of the cycle, understanding DNA synthesis, how it happens, the mechanism. But before we start class, I have a few announcements. First is that there is no lecture on Tuesday, September 30th or Monday, September 29th. As you recall, Tuesday, September 30th is Truth and Reconciliation Day, so it's a non-instructional day. So if Tuesday's class misses class, then Monday's class also has to miss because we want to keep things consistent. But there will be class October 1st and October 2nd for Cycle 4. So Cycle 4 will only have one lecture, and there is a quiz associated with Cycle 4, and that is due October 5th at 11 p.m. There are no labs next week for anyone. Whether you're week 1 or week 2, nobody has labs because, again, there are several hundred students missing lab on the Tuesday, so we can't make up those labs for those hundreds of students. So everybody takes a break for that week with respect to labs. Cycle 3, OWL quiz is due this Sunday at 11 p.m. and Cycle 4 on October 5th. Just some reminders for you. There is an office hour room change for me. My office hours will now take place in Middlesex College, room 105B. It's still every Thursday from 1 to 2:30 p.m. during my lecture cycles, but the room has changed. There was a large turnout and many people did not have a seat, so I decided to change the room with a higher capacity. There is a lost and found in NCB 240, i.e. this building on the second floor, that's the dean's office. If you find anything in this room, or if you've lost anything, please use this room to either deliver lost items or retrieve lost items. We want to help our peers, we want to help each other out. If we forget things, then we want to make sure that that person gets it back. The next thing I want to say, and this comes from me just worrying about you guys, because as I'm aging, I'm worrying more, and I'm a mom, and so I worry about my kids, and so I worry about you guys. So what I have to do is tell you just a brief summary of Cycle 3, begin Cycle 3, Lecture 6. When I say C value or size of the genome, it's the same thing. When I say coefficient of C or copies of the genome, that's the same thing. When I say coefficient of N or ploidy, that's the same thing. When I say N value, that's the number of unique chromosomes. Ploidy is also the number of sets of unique chromosomes. So please understand, on an exam, when I'm asking you very specific things, you understand. When I say genome copy number, you're not thinking of genome size. When I say genome copy number, you're not thinking of ploidy, because you guys know that ploidy doesn't change during the cell cycle, but genome copy number does, right? So please understand and read the question really carefully. None of it is a trick. If it was a trick, I would not be telling you and warning you. So please pay attention to that terminology in my exam. Why are we learning DNA synthesis? When I took DNA synthesis in school and in undergrad, all I wanted to do was just learn it and get my mark. That was the goal, learning for the sake of learning, just to get the GPA. How a tumor grows. Tumor growth is just for the sake of growth. So we don't want to do that, right? If you understand the applications of why you're learning what you're learning, then you retain the information better, you learn with more enthusiasm, and it's just a better quality of your education. So what are the uses of DNA synthesis? DNA synthesis, I thought, was just, okay, here's a strand of DNA, it gets duplicated, now you have 2 copies, or you have double the amount of copies, that's wonderful. So do I have to know all of this stuff? Replication fork, leading strand, lagging strand, all the enzymes involved, and directionality, polarity, all this stuff, just to know that in the end of the day, you get double the amount of DNA. Whoop-de-doo. But as you go through your education, you realize how important this S phase actually is. What are the applications? Well, first, this whole title of this lecture is Inheritance of Sameness. How is it that it's possible for the daughter cells, or the progeny, to be identical to the parent cell? It's possible because of the way replication occurs. It's semi-conservative, and there are complementary base pairs that are being added. So that maintains fidelity or accuracy in copying that genome. that's one reason to even understand how S-phase works. How is fidelity maintained? How is accuracy maintained? How is duplication maintained? How is sameness maintained? Second reason is DNA technology. Whenever you want to study a cell, you have to study the DNA, you have to study the cell function. A lot of it has to do with the genome. You have to use DNA technology, like techniques to manipulate the DNA, techniques to manipulate the cell. It's the only way to learn, right? Learn or to develop new therapies and drugs. So you have to do techniques on them. All techniques, they stem from a natural system. If you understand the natural system, you understand the technique. You can develop new techniques if you understand the natural system really well. And that's something I don't even know when generative AI can even do something like that, to be honest, really, to the point where it actually works in a lab. Like your number one competitor is, your students are your competitors, your peers, but your number one competitor, you guys know who you're competing with, right? So you have to, at least in an interview, be able to shine and be able to say like why it is that you deserve this position, because they want to see, are you a leader, are you a follower, are you a thinker, can you critically think, can you beat AI, that kind of stuff. So you'll be chosen, right? So understand that you need to understand that it starts here. To understand the natural system, everything stems from it. PCR, polymerase chain reaction. If you work in a lab, you're going to use PCR. You have to amplify DNA, whether you want to clone, you want to do any sort of sequencing, anything you want to do, you need to learn how to do PCR, period. DNA sequencing involves DNA synthesis. qPCR is a spin-off of PCR. That's how you detect the presence of viruses in an organism, right? So qPCR research was already quite there because it's very, very accurate, quantitative PCR, but it really soared when COVID-19 hit, right? That's how you test for COVID-19, qPCR. What is the premise of qPCR? Almost everything from start to finish is DNA synthesis. They take the mRNA, convert to DNA, and then they amplify the DNA using synthesis. All of these enzymes that you learn about, all of this, it all involves diagnosing COVID-19. So please understand, you need to understand this stuff. Not just the end result is that the DNA is duplicated. That's boring, right? That's what I was told. That's why I didn't care to learn and pay attention. I had to pay attention later on and go, oh, I should have paid attention in first year. All right, the third reason is drugs. Do you guys know how chemotherapy works? What stage does it target? Or what does chemotherapy target? Chemotherapy is still the #1 treatment, right? Even after 100 years, we're still in surgical resection if needed, chemo and radiation. All that other stuff, that adjuvant therapy, comes later, right? But that's still the primary, first line of treatment. So how does chemo work? Yes? It uses strong chemicals to kill off cancer tumors, yes. It does target rapidly proliferating cells. It's a cytotoxic drug. It kills a cell. But what's the process? How is it killing the cell? What is the cell not able to do that it wants to do? Yes, replicate. What is it targeting in the cell cycle? Chemotherapy targets the S phase, period. There are different classes. One class targets topoisomerase. One class targets by making bonds so that it targets helicase. One class targets, et cetera, et cetera. Once you understand DNA synthesis, you just have to read the classes and what they do, and you're like, boom, I know how this drug works, right? If you don't have the background, you're just like reading, oh, what? What's that? Then you go back to the basics. Then you go back again, you go back. People take forever to learn material, right? But you guys are going to be so smart and so efficient. You're going to pick up articles and learn them because this is the way we want to train you. This is all part of your training. You didn't pay tuition to come here and just diddle-daddle, right? So, DNA synthesis. When I learned this, I learned all of this in 2D. You guys now have YouTube and all kinds of videos that are exciting. I didn't have that, 2D. When I first saw this video, I was in such awe because these are the shapes of your molecules. That's actually the DNA structure. This is happening in real time in your cell. And to think that these molecules, they form this very specific structure, they can conform, they can change conformation, and they can do it at such a rapid rate with such accuracy. These are molecular machines in your body that work for you. This is happening in your body, many of us right now, in our cycling cells. So it's just incredible how it works. It separates the DNA strand really quickly. Helicase is just separating constantly. Those yellow things that you see are bases. And polymerase is reading each one of those and looking at that base and adding the complementary base. It sees an A, it adds a T. It sees a C, it adds a G, and vice versa. With such accuracy. How many bases do you have to replicate in one nucleus? You guys know this, 6.4 billion were diploid. 3.2 is our genome size. 6.4 base pairs is what it has to replicate. And imagine, it does it with such accuracy. Does polymerase make mistakes? It sure does. It does make mistakes. You get mutations from an endogenous system. You endogenously get mutations as you age, because polymerase does make mistakes over time. Something more higher level thinking is some people would say that polymerase purposely makes mistakes so that you can drive evolution. But we have to maintain sameness and difference. Because when polymerase makes mistakes, or when you get mutations from the environment, then you get differences, right? But if you always had sameness, we would never evolve. If you always had difference, that would not be viable, period. If your liver cell divided and just made a different kind of cell each time, your liver would stop functioning. So you have to maintain that balance between sameness and difference for us to be who we are today and to evolve and to be the same. Confirmation. careful what you're watching on YouTube. This is a 2-minute YouTube video. Did you guys find that incredible or not? Maybe it's just me. This is a 2-minute YouTube video that you can watch. Gives you animation of DNA replication. It's a very old video, but I went through many recent videos, and believe it or not, just when you think this video is perfect, there is a mistake. They mention a mistake, and I'm like, oops, that was not correct. So just be careful what you're watching on YouTube. Whenever I feel like you need to pick me up, you don't feel confident enough, you feel like you can't do this, you need someone to give you that push and that motivation, I think of these molecules that are working in my body. No one works as hard for me, so I can't let them down. It sounds really weird, but sometimes it actually works. DNA replication and cell mitosis. So this is mitosis, not meiosis. This is just simply telling you that you have your homologous chromosome here. This is chromosome 1 from mom, for example, and 1 from dad. These are going to duplicate through the process of synthesis, which you'll learn today. And then they line up in the metaphase plate. This looks like crossovers occurring. There's no crossover. They're just lined up. This is in mitosis. And then the cells separate. And the reason these are identical is because each strand here is being used as a template. There's a parent strand and a daughter strand. So a template and a newly synthesized, which is being copied over. So if it's copied over and if it's synthesized the way it is through semi-conservative replication, it's bound to have cells that are identical. So let's go over some of the rules, like i.e., what is the structure of DNA and how does polymerase work? Once you know the rules, there's only one way in which replication can occur. So the first thing, the DNA has distinct 3' and 5' ends on each strand, which confers its polarity or position. So very distinct 3'. This is a 3' and that's a 5'. Here you have your 5-carbon sugar. On the 5th carbon, you see the phosphate group. That's why it's called 5'. And on the 3rd carbon, in that sugar, you see the 3' OH. 3' OH. Polymerase needs to see that in order to extend the DNA. So it needs a pre-existing nucleotide to extend. It can't take a base and stick it onto thin air. It needs to attach it to something that's already pre-existing, and that's why it needs a primer. That's why it needs a primer. So that's why you have that OH group that's really important. It has that free phosphate group, and the 2 strands are anti-parallel. So whenever you draw the DNA, it's always anti-parallel. One end is 3', the other end will be 5, and vice versa. And 3 and 5 go together, not 5 and 5, and 3 and 3. This right here is the backbone of the DNA. You have all these sugar groups that are attached by these phosphodiester bonds, and that forms the backbone of your DNA. These are your nitrogenous bases. They're also called DNTPs. N is for any nucleotide, so ATP, TTP, CTP, GTP. These are complementary base pairing with each other, triple bond for CG, double bond for AT. The reason they pair in this fashion is because C and G fit perfectly in the pocket of that helix. A and T fit perfectly. If you get C and A pairing, that's a mismatch or a DNA damage, then what happens is you get a distortion in the backbone, and that is then recognized by repair enzymes. P53 can also recognize that distortion, and it would send repair enzymes there and say, look, there's a distortion in the backbone, there must be a mispairing. It's not a Watson-Crick base pairing. Replication is semi-conservative, meaning that each strand of the double helix serves as template, meaning each strand serves as something that polymerase will use to copy over. I'm not going to say parent and daughter. I only see that in a first-year textbook. It's always template and new strand. Okay, so if you go into a lab and say the parent strand, people are going to, yeah, they're in first year. Okay, so it's like just say template. So the template is going to serve as that copy, going to now copy over that template. And that's why you get identical cells. Here you can see, this is a figure right out of your textbook. You have your template and then the new strand. The new strand is being made. The 3' OH is required because that is what polymerase recognizes to then go and add the next base. The next base in this case would be an adenine because it sees a T. It sees a T and it goes and adds an A. And you guys saw how fast it does it, right? In that YouTube video, it was incredible how fast it did it. So it gets its energy, this is a very high energy demanding process. It gets its energy from this triple bond P. When the bond breaks, it releases energy, just the way you know, you release energy from ATP, right? Then you would get the energy you need to form this bond and all of these bonds here. This is an energy demanding process. This right here is DNA right now. I mean, this is they're saying it's a new strand, but this is why polymerase needs a primer, because it needs a pre-existing nucleotide. What's a nucleotide? It's RNA or DNA. That's a nucleotide. What's the main difference between RNA and DNA? RNA has uracil, DNA has thymine. You should know that basic difference. There's one difference I want you to know. It should be that. So the primer in our body doing DNA synthesis is an RNA molecule. It's not a DNA molecule. It's RNA. And so that RNA then has to be degraded, and then DNA needs to be synthesized. Replisomes are all the enzymes that come together to synthesize that portion of the DNA. So all the enzymes, all the proteins, everything that comes together. Here you have your topoisomerase. Here you have your helicase. You have this clamp that keeps DNA polymerase in position. There are other proteins as well that are involved in here, like single-stranded binding proteins, but that are not shown here. You don't have to know what's not shown here. Replisomes, they replicate one strand continuously and one discontinuously. So there's one leading and one lagging. This is a replication fork. Probably the only thing you have to memorize is this here. DNA polymerase synthesizes in the 5 to 3 direction using the 3 to 5 template. So it reads the template. This is important in every exam question you'll do for these types of questions. It reads the template 3 to 5, but it is making the new strand 5 to 3, which makes sense because these have to be anti-parallel. They have to be anti-parallel. So it's not going to make it 3 to 5 if it's reading it also 3 to 5. It's not going to make it 5 to 3 if it's reading it 5 to 3. So it reads it 3 to 5, makes it 5 to 3. If you put all that information that I gave you about the characteristics of a DNA molecule, what a DNA looks like, why it is the way it is, and what polymerase does, how it reads, and which direction it goes in, there's only one way in which DNA replication can occur. You would design it this way too, and it's just pure logic. This is not memorization. So all you really have to know is what a DNA looks like and what polymerase can do. And you will never have to memorize this down the road. What was that again? Leading, lagging? I don't even understand. Just understand these couple of things, and you can always come up with it using your intelligence. So chemotherapy, one class is topoisomerase inhibitors. They come and they literally inhibit topoisomerase. What does topoisomerase do? It relieves the tension. Imagine that helicase is unwinding that DNA, taking 2 ropes and just pulling it apart. Eventually, you won't be able to pull it because there's tension there now. So what topoisomerase does is it cleaves or cuts and it rejoins the double helix. Cut, rejoin, and it keeps doing that to relieve that tension to allow helicase to do its job and synthesis to occur. There's another class that forms a bond between the 2 strands such that helicase can't unwind the DNA. So now synthesis can occur. There's another class of chemotherapeutic drugs that targets the enzyme that makes the DNTPs. Enzymes are required to make DNTPs. So the cell's ready to replicate, but they're like, we don't have any bases to put on. We can't put on the complementary bases. So synthesis stops and the cell dies, the cancer cell dies. So Understanding this has a lot of application, guys, everywhere, from technology to whatnot. Yes? That's a good question. Does it target everything? You guys know chemotherapy is given systemically, so it targets rapidly proliferating cells. And this is why it targets a lot of your gut cells that are regenerating, your hair follicles, cells that rapidly proliferate. Now, you learned about the fork, right? How many of you guys did the textbook reading for this cycle? It's very important. Good job. It's really important. I'm going to lecture assuming that you read it all. So what's a replication bubble? What's a replication bubble? A bubble is two forks put together. A bubble is where replication originates or starts. It's called the origin of replication. I will refer to it as ORI. I've only seen it written as origin in a first-year textbook, so ORI. ORI is where the replication starts. Why do you think it's a bubble? Why is it not one fork? Yes? Very good, excellent, perfect answer. If it were just one fork, replication would take twice as long. One starts this end, the other one starts this end, and says, you know what, let's be efficient, let's just meet up. You start on that end, I start on this end. We have two forks, i.e. makes a bubble, and then we're gonna go ahead and meet up. This is a prokaryotic cell, so usually there's just one replication bubble. But in linear chromosomes, large linear chromosomes, boom, 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 multiple bubbles are fired. So this is a generic replication bubble, showing you the origin of replication, and how it has leading and lagging strands, where the primers are, what Okazaki fragments are, and et cetera, the polarity, the directionality, et cetera. This is right from your textbook, so my understanding is that you've read it, but let's just quickly go over this. The blue are primers, and the red is DNA. One strand on top here is leading, the other one is lagging. At the bottom, you also have a leading and a lagging. If a replication fork has a leading and a lagging, then a bubble obviously will have two leadings and two lagging. But keep in mind that they're crisscrossed. Don't put both the leadings on this side and both the lagging on that side. If the leading's on the top right, then the other leading is on the bottom left. So just crisscross them. What's happening here? I think it's really important to understand what exactly is going on. Here you have 3' end of the primer. That's the 3' end of the primer. That's the 3' OH. That polymerase 3 is using to synthesize. You have to know the difference between polymerase 3 and 1. Then what happens is polymerase 1 will come, it will remove this primer, and then it will synthesize the remaining. You don't have chunks of RNA in your DNA, right? RNA has to be removed, synthesized by DNA. Same thing here. This is the 3' end of the primer, 3' end of the primer. And what is polymerase 1 doing? So polymerase 1 is going to come and it's going to remove that primer. That's the job of polymerase 1. And then it's going to synthesize all of that. What is it using as its pre-existing nucleotide? It is using the Okazaki fragment, 3' OH. That's what it latches onto, and it continues this way. When this Okazaki fragment here and this meet right there, that is where ligase seals the nick. Number one mistake on this type of question is, where does ligase act? Students will say, oh, the ligase acts here, or it acts here, or it acts here. It's a 1 in 3 chance of you getting it right if you're guessing, but don't guess. It can't act here because it's not required. Ligase just seals the nick. Polymerase is taking care of this. It's tacking it on. It's tacking on the bases. It's chucking along. This primer, ligase is not going to seal a primer. The primer has to get out. So that, nothing, out. It's when the two Okazaki fragments meet, that's where ligase acts. If you understand that, you will get all the questions right when it comes to ligase. Number one mistake. Linear eukaryotic chromosomes, they fire several origins simultaneously. They have to. Look at how long our chromosomes are. Imagine if one ORI was like, okay, I'm doing this. No, multiple ORI, they fire at once. and that chromosome gets duplicated pretty fast. This is what ori actually look like. It's an electron micrograph showing multiple replication bubbles of the Drosophila melanogaster, which is a common fruit fly. This is what the ori actually looks like. There's an example here, an example here. That whole thing is an ori. That's an ori. So, you know, when you look at these textbook cartoon pictures, they have this perfect bubble, the perfect circle. I want you to go into upper year or a lab in your summer job or anywhere, and if you look at something like this, I want you to be like, yeah, I'm pretty well trained. You know, I can recognize an ori. Now, what happens at the very last or end of the chromosome, i.e. the last bubble takes it all the way to the end, right? What happens at the very, something very critical happens at the end. And this will explain, guys, cancer. It'll explain why it is that cells senesce, why it is that cells die, lifespan. There's a lot of information at the end of the chromosome. This is a really important concept. So over here, what you have is the end of the chromosome. Notice this, the leading strand is perfect, meaning it is synthesizing and copying till the end of the template, right? What's the problem in the lagging strand? Focus on the red DNA, because the primer will get removed. Did you guys notice that the newly synthesized strand will be shorter? Yeah, it'll be shorter because this ultimately has to get removed. That can be part of your DNA. In the next replication round, guys, the red are the ones that are going to serve as template. You're going to get separation, and then the red will be one of the template. So if that's the template, in the next replication round, it's even going to get shorter, and then shorter, and then shorter, and shorter, and shorter. So as your DNA replicates, the ends of the chromosomes get shorter and shorter and shorter. They eventually will hit a critical point where they're going to now start to affect genes that code for important proteins or important RNA. So what do we have at the end of our chromosomes to buffer and to protect our genes from being mutated? They're called telomeres. Telomeres are just repeated sequences, TTA, GGG, TTA, GGG, repeated many times to say, look, you are going to shorten the chromosome. That's a given. So why don't you just shorten a non-coding region? Shorten something that doesn't code for anything, doesn't affect your genes. It's like the caps on your shoelace. They protect the shoelace from fraying. So if you cut the cap over and over and over again, you reach a critical point where now if you make one more cut, boom, the shoelace will fray. So we don't want that to happen, i.e. we do not want the genes to be affected. So here's my question. What does polymerase need to fill in that gap? Now you might be saying, it's so easy, take your pen and just do that. Take your red pen, Nikki, and just do that. Can't polymerase just come and hop in here and just synthesize that part? No, it can't. It can't because it needs a primer. It can't just stick on a base there, and it can't go this way. So it's limited. It's limited by its evolutionary past. It's restricted. So whenever you ask yourself throughout your life, why can't this enzyme do this? What's wrong with it? Nothing's wrong with it. It's just restricted through its evolutionary past. That's just the way it functions. Why hasn't it evolved to do this? Maybe it will later on, or maybe it hasn't for a purpose, for a really good reason. Like you can't take a Honda Accord and turn it into a Lamborghini. It's just restricted. So you can maybe change out its engine, change out its tires. I don't know what it takes to change that to a Lamborghini. Impossible, but you can make it closer, right, through time. But it's restricted. So what has evolved to maintain the telomere length, so do all our cells maintain telomere length? Nope. Somatic cells in your body, meaning all the cells in your body except for the stem cells and your germ cells, your gametes, they are somatic. So liver, lung, eye, hair, everything, somatic. Somatic cells do not express this enzyme that can extend that template. What's that enzyme? It's called telomerase. What does DNA polymerase need? It needs more template, guys. It's all it needs. It just needs more template. Once it has more template, then primase is going to come and add the primer. There you go. That's the 3' OH. That's 5'. It's just going to add that primer, and then DNA polymerase III is going to come and go, oh, got it. It's going to synthesize, and look at that. That area has been restored. So the enzyme that extends this template is called telomerase. We don't have telomerase in our somatic cells, but we have telomerase in our adult stem cells and our germline. So that brings us to telomerase and the Hayflick limit. Here you have the telomere in your chromosomal DNA. After each replication round, this is shortening and shortening and shortening until it reaches critical length. Once it reaches a critical length, it has reached its Hayflick limit. And once it reaches the Hayflick limit, the cell senesces. So cell senescence means that it will no longer replicate. It is not going to replicate. It was not going to go into G2M cycle. So a senesced cell can function. Senesced cells can actually do their function. They just won't replicate. They can arrest in G1 and remain arrested in G1, or they can apoptose. One of those three things can happen depending on the tissue and depending on the environment, depending on the situation. So yeah, cell senescence. Our cells can replicate, our somatic cells can replicate anywhere from 60 to 70 times until they reach senescence. Not all of them do, but those that do replicate 60 to 70, and that's why you have your adult stem cells to go and regenerate. Here is an example of cells that do express telomerase, where you get the shortening of the telomere, but then the telomerase will restore the length. So which cells are those? Well, our adult stem cells have them. What do our adult stem cells do? They regenerate your tissue. You can't regenerate a tissue by giving it old DNA, by saying, oh, I'm going to give you a brand new cell with decreased telomere length. No, I'm going to give you a brand new cell with lengthened telomeres, and then you can drive it down and get it to Hayflick limit, and then I'll regenerate it again. That's what these stem cells need telomerase. Germ cells, you know, the germ cells that make your gametes, they also have telomerase, but the sperm only. The oocyte doesn't have telomerase because the oocyte doesn't replicate, but the sperm does. It does need telomerase. However, after the zygote is formed, you get fertilization and you get the production of the pronucleus, then the gene in the oocyte of telomerase gets turned on. The telomerase gene in the oocyte will get turned on. Cancer. Do you think cancer cells can grow if they're restricted by the Hayflick limit? The cells will senesce, it'll undergo apoptosis. Cancer cells know the only way to replicate is to enter the cell cycle, but they know they're limited in the number of times they can replicate. So they must activate telomerase. You had telomerase when you were a baby. When you were a fetus, you had telomerase. So embryonic stem cells, hands down, have telomerase. They can't replicate 70 times to make an entire baby. They need to replicate billions of times. So they need to have lengthened telomeres. So yes, telomerase is highly active during embryogenesis. So you have that gene in your somatic cells. So when you get liver cancer, the telomerase in your liver cells are turned on by cancer cells because they need to maintain the telomere length. So here you have the Hayflick limit. This is what it means. You basically have reached that critical point. Cell senescence is irreversible cell cycle arrest, no longer in the cell cycle, and the Hayflick limit is the number of times the cell divides before cell division stops. And that's roughly 60 to 70 in our somatic cells. Here's a graph that I would usually just give you on an exam and just tell you to deduce this yourself. But in the past, I've done that, and it hasn't gone over so well, so I'm just giving you a little bit of a heads up of how I want you to think of some questions. On the x-axis, you have number of cell doublings, and on the y-axis, you have the telomere length. Whenever you see a graph on an exam, students panic because it's a new graph. They've never seen it before. It's an application question. I'm asking you to take the knowledge that you already know from lecture and to apply it. The nervousness starts there. I can tell. The stats prove it because students generally don't do well on those types of questions because they're nervous, I think. You know what they failed to do? They failed to read the x and y axis. If you just read what it's saying, you can actually answer a multiple choice question and go, oh, that makes sense to me. answer B makes sense based on X and Y. So just say, what is X telling me? X is telling me that it's the number of cell doubling. So the cell is doubling, meaning the cell replication is increasing as I go from left to right. What's Y telling me? Y is telling me that as I go higher and higher, the telomere length is increasing, and as I go lower and lower, the telomere length is decreasing. So once you know that, you can pretty much figure out what this graph is saying. The red represents somatic cells, and so this makes sense. We don't have telomerase in there, so as the cell doubles or divides, you're going to see a decrease in the telomere length, right? That makes sense. Adult stem cells have telomerase, but not to the capacity of the embryonic stem cells in the male germline, but they do have it. So telomere length does decrease, but not at that rate of a somatic cell. It decreases just enough that it's good for regeneration of your tissues. Embryonic stem cells, those that have to undergo embryogenesis, right? The entire embryo has to develop. They absolutely have to have telomerase. And the male germline, the sperm, has to divide over and over and over again. You can't pass on decreased telomere to your offspring. Well, sorry guys, it's the best I could do. They're decreased. This is what you have to give. You have to give it fully lengthened telomeres to your offspring. And that's, they need a good start. So the zygote will then pick up, and it will continue with the telomerase. Now let's focus on the red here, the somatic cells. Look at this, boom, boom, boom. Okay, reach cell senescence, Hayflick limit is right here. But when there is instability in the genome, meaning the genome is not stable, what does that mean? Genome instability is something you'll learn about in upper year. It just means that something is not right in the genome. There are mutations in genes. We can't pinpoint exactly where they are unless we sequence them and do a lot of research, but there's some instability in the genome. Some mutations have happened that the cell is now pushing through senescence. It's not arresting in G1, it's not undergoing apoptosis, and it's going into the replicative cycle, i.e. it's doubling. But you're like, but you said the cell senescence cells don't divide. They don't enter G2M and S. But in this case, they're entering. So there's something wrong with them, right? They push through senescence, that's a bad thing. Because now you're reaching crisis. When you reach crisis, the cell's pretty much done. There's really nothing you can do unless you want to get therapy. Boom, all of a sudden now, after crisis, you start to see an increase in the telomere length. And now this becomes a cancer cell. What do you think happened right there? If I were to ask you this question on the exam, what event do you think, or a plausible event that took place here? Yes? Perfect. Telomerase was activated here. The cancer cells said, okay, we pushed through senescence, we got there, because we didn't apoptose, thank God. Whew. But now we have to get telomerase. They probably mutated P53 at this point, too. Not all cancer cells have P53 mutated, but there's a really good chance. So telomerase, what is it? It's an RNA-dependent DNA polymerase. You guys heard of this in cycle 1, an RNA-dependent DNA polymerase? We have that too, telomerase. It uses RNA to make DNA. It also needs a template because that's the way it evolved. It can only extend the template by adding complementary base pairs next to it. It can't just take the next one and go, oh, I'm going to add a T, and another T, and a G, and a C. It can't do that out of thin air. It also needs to copy, and it needs to do complementary. But it comes with its own template. So this might be confusing. Template to make a template. Yeah, it needs its own template to make more template for the DNA. It has its own repeat. And that repeat then is going to make TTAGGG. So AAUCCC is complementary to TTAGGG. So there you go. It increases the length of the template. Now primates will come and add that. Someone, by the way, previous year noticed that this is a typo in the textbook. That should not be a T. That's an RNA. That should be a U. There's a typo in the textbook. A student a couple years ago picked it out and said, why is there a T in there? Isn't that an RNA primer? Boom, you're right. You have to make a correction in the next edition. If you guys find mistakes, let me know. All right, so DNA polymerase, it comes and it synthesizes that part. Ligase comes and seals that nick, and you are done. Look, there's no ligase here. there's no ligase somewhere in the middle here. Ligase is when these two come together. That's where you get ligase. This is an image right out of your textbook, so please look at the summary and understand it. So what has happened here? Basically, what's happened is that you restored the length, right? What end does telomerase work in? What end? You should know this. If I ask you in an exam, point to the end where you would see telomerase. And I'm going to point different things and go, A is pointing to this, B is pointing to this, C and D. Where would telomerase be working? It's not a guessing game. Don't guess. But if you must memorize, telomerase works on the 3' end of the template. And there is one end here. These are linear chromosomes, so don't forget about the other end of the chromosome. Here and here is where you would see the action of telomerase. If you memorize it, it's fine. If I reword the question, you won't get it. So please don't memorize it, understand why. Because this is being synthesized this way, this must be the lagging strand. It's the lagging strand that gets affected, not the leading. The leading is fine. We have one more slide after this, so please don't leave. What I want you to do is to write down the mechanisms that you've learned that ensures sameness. How do we get sameness? It has to do with replication. I think I already talked about it, right? The semi-conservative, the base pairs, etc. But the answers are posted on OWL. Just go ahead and read them, please. What I'm going to do next is just briefly go over a few things that could come up on the exam. This is just me worrying about you and trying to take care of you here. All right. Ori, if I gave you this on the exam, could you fill out the rest of this bubble? Is that information enough to fill out the entire bubble? Or would you say, nope, I need more information. You haven't given me enough information. So how many say there's enough information here? Okay. How many say, no, there's not enough information, you need to give us more information? Oh, everyone agrees there's enough information. Awesome, perfect, absolutely. Apply the rules. If that's 3', this has to be 5'. Anti-parallel, 3', 5'. Draw that out. Now, can someone tell me which 2 strands are leading and which 2 strands are lagging? Yes? Top left and the bottom right are leading, okay? I just want to know, agree, disagree. Does the most of the class agree? There are not a lot of votes. How many say no? Okay, so no, people aren't raising their hands regardless, okay? Are you guys going to guess? Come on, guys. Okay, we got 4 minutes. You got this. Oh my God, I'm so glad I'm doing this for you. Yes?
I think it's still my original answer because the DNA polymerase can only replicate 5' to 3', it has to read the strand 3' to 5.
You're right. It is reading at 3 to 5. You're absolutely right. It is reading at 3 to 5. So is all of this top part then all leading? No. So you said the top left is leading. Yes. Okay. Yes. From the Ori, the top to the right is leading. All right? Because guys, if it's Ori is where it starts. People are congregating at the Ori. People are congregating at the Ori going, okay, here's where we start. They're not congregating at the 3' end on the left. That's not where the congregation's happening, right? Congregation's happening and the Ori, origin of replication. So the primer is going to go there. Where's the other primer? It's got to go the other way, guys. A one-way street. So one-way street, the other primers go here and here, right? Beautiful. Now, can you guys label the 5' and 3' ends of the primer? You can do it. This is 5', this is 3'. 2 ways to know it. It needs a 3' OH. 3' OH. Well, how the heck is it going to go the other way? It's going to go that way, right? So that's the other way to look at it, guys. Look, 5 and 3 go together. 5 and 3, anti-parallel. Make the primers anti-parallel as well. 5 and 3 go together. Beautiful. Okay, so this is 3. I point to these things on the exam and I ask you, is this the 3' end? Is this the 5' end? I'm doing this because this is common mistakes. All right, we have 1 more minute. Polymerase 3 is going to come and do this, 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 right? Does that make sense so far? Good. Okay, now, what's polymerase 1 going to come and do? Polymerase 1 is going to come and it's going to erase this primer, all of them, but we'll just do this one as an example. And then what's going to happen? Polymerase 1 will also go and synthesize that gap. That's polymerase 1. That's polymerase 3. And where does ligase work? Where is ligase? Tell me, guys, is ligase here, 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 here? Where would ligase work? Here? How many say here? Okay, we've got a couple people. No, guys, this is the 3' OH. This is the 3' OH of the Okazaki fragment, which polymerase is using to extend. That's its quote-unquote primer. So ligase has no business there. It doesn't need to do anything. But look at this. The 2 Okazakis meet. Boom. That is ligase. Okay, if you ever get confused, memorize this. Ligase acts at the 3' end of the primer. It has nothing to do with the primer. The primer gets removed, but if you really want to know the location, it's a 3' end of the primer. Last thing, guys, last thing. Does ligase act in the leading strand or only the lagging strand? No, no, ligase, ligase. What's happening here? What's happening here? Ligase. All the bubbles in the leading throughout the chromosome. Ligase acts in both strands. Number 1 mistake, okay? All right, so hope that helped. Take care, guys.